So in just the previous session, we completed this part as how the uh, activation of a B cell is done through the three signals, and thereafter how the B lymphocytes are proliferated and differentiated. Now coming to one important aspect of immunological tolerance is the concept of receptor editing. Just as I was saying that whenever there is a membrane bound immunoglobulin or there is a soluble immunoglobulin, so this is an immature immunoglobulin. So if you can see the picture, these are the bone marrow stromal cells, and the IgM, the membrane bound IgM is partially activated. This is, again I'm saying, this is an immature immunoglobulin. And if this immature immunoglobulin molecule is self-reactive, that is, it's overreactive to a host antigen, then what happens? There is a phenomena that occurs, that is the receptor editing. So in case of receptor editing, as I said, if immature B cells recognize self-antigens that are present at high concentration in the bone marrow, and especially if the antigen is displayed in multivalent form, see, the antigen is displayed in the multivalent form, that is on cell surfaces, okay? Many antigen receptors on each B cell are cross-linked. So if the antigen is present on this multivalent form, many receptors on B cell receptors cross-linked. One consequence of such signaling is that the B cells reactivate their RAG1 and RAG2 genes, if you remember them from recombination, and initiate a new round of VJ recombination in the immunoglobulin kappa light chain gene locus. A V segment upstream of the already arranged VJ unit is joined to a downstream J segment. So if you remember the VDJ or the VJ joining, the V joins with a J. So that has been done. However, if there is a self-reactive antigen, the fresh rearrangement is occurring. Another V segment of the kappa light chain joins with the J segment of the kappa light chain. As a result, the previously rearranged V kappa and J kappa exon in the self-reactive immature B cell is deleted, and a new immunoglobulin light chain is expressed, thus creating a B cell receptor with a new specificity. This process is called receptor editing and is an important mechanism for eliminating self-reactivity from the mature B cell repertoire. Now coming to, this is a very simplified structure of the B cell receptor. I'm not getting into the details of the B cell receptor. And these are the structures of the two polymeric immunoglobulins that are produced. And they are also produced in the secretory form. They have the secretory variant as well as the membrane bound variant. Now this is the movement of dimeric IgA. So this is the secretory IgA, and this is the IgA that is to be secreted. And as I was saying, that some cells are capable of transferring a dimeric IgA through the transport vesicle to the mucosal lumen. If you remember, when we studied the structure of Golgi yesterday, I briefly had showed you that there were the presence of secretory IgA molecule. The secretory IgA molecule has a blue color chain, which is actually the blue color chain. With the blue color chain, the secretory Ig molecule is released and enzymatically cleaved. So this is the secretory component of the secretory IgA, and within this component they are released, so that this secretory component gives this immunoglobulin A protection from the acidic environment of the lumen of the body tract. Most lumen of the body tract have acidic environment. Now coming to another important thing I just wanted to show you, if you look into this structure, there are two diagrams here. The first diagram is showing the IgM and the IgD co-expression. So we have a rearranged heavy chain. These are the primary transcripts, and you can see there are two polyadenylation signals. One after the C mu and constant region mu, and the other after the constant region delta. There is differential process. So these are two polyadenylation signals. So after the differential processing, we are getting two different RNA transcripts, and then RNA transcripts are further spliced to give me a mu chain and a delta chain, which are subsequently co-expressed in a B lymphocyte. Okay, and this is this is a picture of the expression of membrane-bound immunoglobulin and a secreted immunoglobulin. If you look into this picture, you can see that behind the mu segment is SE. SE is the tail piece. So this tail piece is present. So we have a SE segment. And in this there is a polyadenylation, and after the SE segment, we have two segments, M1 and M2. Now, the secretory piece or the tail piece is an additional piece for IgA. It's not found in IgM. It's found in IgA. And this piece is added in the lining epithelial. This piece is added in the lining epithelial cells of cavities as it is transported across to lumen. The secretory piece protects the antibody from proteolytic digestion. So that is the main role of the secretory piece. Now, let's check this one. So we have a polyadenylation site and after SE, that's the tail piece, and we have a polyadenylation site after M2. So this is the primary transcript. And there is differential expression. We are having two transcripts. So this is, and then we are having RNA splicing. So if you can see here, after C mu, M1 and M2 are joining with the splicing out of the SE segment. So we are getting the membrane-bound IgM. But in case of secreted IgM, if you look into this, we are having another polyadenylation signal after the tail piece or the secretory piece. And when the intermediate segment is spliced out, so the joining results in the production of a segment, mRNA mu with tail piece, and so it's a IgM with the tail piece. Sorry, tail piece is present both in IgA and IgM. That was my mistake. So we have a secretory IgM molecule with a tail piece. So it's the same thing, just there's an alternative splicing occurring in the IgH locus. So this is an example that has been shown with the mouse immunoglobulin heavy chain locus. Now coming to another part, another important part, since we are right now doing recombination areas of B lymphocytes. Now we had studied the concept of somatic hypermutation and affinity maturation. Now let's look into this. So these are the heavy and the light chain V exons. The heavy chain has V segment, J segment, and K segment. So these are the V segment exons. And there is the 3 CDR1, complementary determining region 1, region 2, region 3. And by the help of these three regions, it is actually recognizing the antigen. What happens? There's a sudden mutation in CDR2 and CDR3. If this mutation is favoring the further association of the antibody with the antigen, the mutation stays. If the mutation is favoring a further, more stronger attachment of the antigen with the antibody, the mutation stays. Further down the line, so this is early primary response. Now, as the antigen is constantly being in association with the antigen, and the epitope is being constantly in association with the CD complementary determining region of the antibody, further mutations are brought in. So this is the late responses, and further mutations, it is seen that the antibody and the antigen are becoming, the association is becoming gradually stronger. So this is the late primary response, and this is the secondary response. As you can see, mutations are accumulating. So successive rounds of mutations are accumulating in such a manner that the antibody is gradually having a stronger and a stronger association with the antigen. So these mutations are selected, and this phenomena is known as somatic hypermutation. And as a result of this somatic accumulation of this somatic hypermutation, you can see that the affinity of the antigen has increased many-fold for the antibody. So the antibody has now many-fold increased affinity for the antigen, and this is known as affinity maturation. So the affinity of the antibody for the epitope has increased as a result of successive amounts of accumulation of somatic mutations. So that is somatic hypermutation and affinity maturation. And now coming to the concept of junctional diversity. The right picture shows the concept of junctional diversity. So if you remember that junctional diversity arises when the fusion of V, D and J segments is not exact. So the joining is not exact. The joining is not exact. Such imprecise joining of gene segments can result from the deletion of nucleotides in the joined region or the addition of so-called P or N nucleotides during VDJ recombination. In about 80% of antibodies in adult humans show this type of junctional diversities. So we have, this is no junctional diversity. You can see the joining is exact, but we can have deletions as a result of which new segments, so there's addition, so there is a TA portion is deleted out. So you can see the junction is differing from the original junction. We might have addition of nucleotides. So you can see again the junction is different from the original junction. Or we may have addition of new nucleotides. So again the junction is different from the original junction. So these are the examples of examples by which the junctional diversity increases when the joining between the V, D and G segments are not precise. Okay, so we are done with B lymphocytes. Now we move on to T lymphocytes. That's another very important cell of the adaptive immunity. If you look into the picture, the upper panel picture is very self-sufficient picture. As you can see that we have a common lymphoid precursor cell. From the common lymphoid precursor cells, we are having the double negative 1 cell, 2, 3. Now just as I said in the first class of this comprehensive summary, that if you remember, the lymphoid precursors have basically two committed subpopulations. One is the T lymphocyte and one is the B lymphocyte. Of the T lymphocyte lineage, we have the alpha beta T cells, we have the gamma delta T cells, and we have the natural killer T lymphocytes. So you can see that the pathway in where the gamma delta T cells are coming out and the point where the natural killer T cells are coming out. So from a double negative 3 stage, when the cell moves to the double negative 4 stage, it is during this tenure that the gamma delta T cell branches out. So this is the normal alpha beta pathway. We have double positive cells, that is cells are positive for both CD4 and CD8. Then single positive cells, there is a positive selection and cell is either positive for a CD4 molecule or a CD8 molecule. And finally, we have two committed members, CD4 positive T lymphocytes and CD8 positive T lymphocytes. However, if you look into this picture, you can see it is from the double positive cells that the NKT pathway is diverging out. So I'll be dealing a bit with the divergence of these two pathways later on. Let's look into this picture. As you can see, this is almost the similar picture, but the rearrangement of the chains have been shown. If you remember, the TCR is made up of two chains. The TCR that we commonly read is made up of two chains, the alpha and the beta chain. Usually, the beta chain is made first. It is made during the double negative 2 to the double negative 3 stage, and the alpha chain. So you remember, see, once the pathway has diverged, so there is beta selection. Beta chain is selected and the pathway has diverged. So once the pathway has diverged, then only we get the TCR alpha gene rearrangement, and then after the TCR alpha gene rearrangement, we get the CD4 positive TCR alpha beta gene, CD8 positive TCR alpha beta gene. But we must always keep in mind that it is under the influence of particular cytokines. It's under the influence of particular cytokines and intercellular contacts that the common lymphoid precursors pass through the 4 stages from double negative 1 to double negative 4th stage. If you see the picture, there is a pre-TCR checkpoint. This pre-TCR checkpoint marks the end of the double negative phase and entry into the double positive phase. After passing through the TCR alpha beta checkpoint, the developing thymocytes will always enter a single positive phase and become CD4 positive CD8 minus or CD4 minus CD8 plus. So this was the basics of TCR development, the T lymphocyte development. Further moving on to the structure of T lymphocyte development, as you can see, this is the thymic microenvironment. Usually the common lymphocyte precursor enters at the junction corticomedullary junction. It migrates through the 4 stages just now we discussed to form the double positive, and when the double positive, this is the double positive cells, and once the double positive cells gradually starts moving towards the medulla, and in the medulla we only get the single positive cells. You can see that schooling are being done by the thymic dendritic cells. This is the stage where the mature dendritic cells school the CD4 positive T cells, and in case of certain immune responses, the CD4 positive T cells, a particular clone may be selected. The T helper 0 cells can be activated, and these are the 4 different basic. There are a bit more variants, but I'm showing you the basic 4 variants. So we have the 4 basic variants of the T helper 0 cells, helper 1, 2, 17, and T regulatory cells. I'm not getting into details of these cells because we have discussed the details of these cells earlier in our classes. These are some of the important co-receptors. I mean to say the I will not say the co-receptor. These are some of the important adhesion contacts that occurs between the T lymphocytes and the dendritic cells. So this is the first signal. That's the main signal. And the moment the signal is received, the 3 more important adhesion contacts are mediated by the T cells are the LFA1, then via the CD2 and via the ICAM3. So these are some adhesion contacts that are mediated by the TCR and the dendritic cells, so that the T cells are further primed. And coming on to the different molecules. So this picture actually shows the different molecules that play a big role in selection of any double negative population. And as I discussed just 2 slides back, that we have a pre-TCR checkpoint, and once that pre-TCR checkpoint crossed, the double negative 4 is now committed to become a double positive, and we have the single positive lineages. During the passage through the thymus and through the double negative stages, followed by the double positive stages, if you remember, there were 2 categories of selection done. One was by selection by neglect, insufficient TCR signaling, that is death, positive selection, so weak TCR signaling. Just keep in mind that whenever I'm talking about signaling and activation of a cell, initially when yesterday we talked about the activation and the signaling that is perceived by a cell, for a cell to be activated properly, the signal should be weak. Just we did the case of B cell receptor editing. If you remember, if the signal was strong, then there was receptor editing. If the signal is weak, so the cell is actually activated. So whenever there is an activating signal, it should be kept in mind that the initial signals should always be weak. if there is a strong signal that always results in case of B lymphocytes into receptor editing, followed by, if then also the signals are stronger, apoptosis. And here in the picture, it is very much evident that if it's weak signaling, then only survival of the immunocytes, a single positive immunocytes is possible. However, so this is positive selection. However, if the signals are strong enough, then as you can see from the picture, this is negative negative selection, and the cells are forced to die an apoptotic death. So this was about the alpha beta T cell receptors. Now, this is another picture which shows us the function of the variety of the T lymphocytes. So this is the T helper 1 cell activation, and this is the T cytotoxic cell activation, as you can see from the two pictures. Now, if you look into the picture, as you can see, for naive T helper cells, so this is the first, the left-hand side is the activation of the naive T helper cells, the T cytotoxic cells, activation, and the CTL's generation of cytotoxicity. We had studied the activation of the three signal mechanism of T cells, in which, if you remember, the naive T helper cells on binding of the TCR2 antigenic peptides displayed on MAC class 2 delivers signal 1 to the T helper nucleus. So this was signal 1 to the T helper nucleus, and also upregulates CD40 ligand on the T helper surface. So upregulation of CD40 ligand, by the help of which now it associates with the B lymphocytes. The binding of the, now look at this picture, the binding of the CD40 ligand further stimulates the expression of B7 molecules. So that's signal 2. Stimulates the further expression of the B7 molecules. So now the co-stimulatory molecule B7 has been expressed. Signal 2 of the T helper nucleus is delivered when these B7 molecules interact with co-stimulatory CD28 molecules on the T helper cells. So B7 CD28 interaction gives us the signal 2 of T helper cells. Mind it, for a B lymphocyte, CD40-40L is the signal 2. In case of a B lymphocyte. In case of a T lymphocyte, signal 2 is the B7-CD28 interaction. Okay, so that's the difference of signal 2 between a T lymphocyte and a B lymphocyte. Okay, so once this signal 2 has taken place, expression of cytokine receptors is upregulated, and the T helper commences production of interleukin 2. So IL-2, so this signal 3. So signal 3 is delivered when the T helper cells interleukin 2 receptors are engaged by interleukin 2. The dendritic cell, which is now, this is a very important word, interesting word, the dendritic cell, which is now considered to be licensed. So the dendritic cell is now licensed due to its upregulated B7 expression, detaches from the T helper cell. The activated T helper cell proliferates and generates daughter cells that differentiate into T helper cells, that differentiate into further T helper effector cells. So once this interaction has happened, so we are getting an highly expressed, activated, licensed dendritic cell. So the license of this dendritic cell has been done with respect to this antigen. Now coming to the naive T cytotoxic T cell activation. A naive cytotoxic T cell activation signal 1 is when its TCR binds with the PMHC1 complex. So this is signal 1, that's common. And if you look here, then we have a signal 2. So the signal 2 is also seen for the naive T cytotoxic cells via the B7 CD28. However, the picture itself also speaks that the co-stimulation in this case is mediated even in the absence of CD40-CD40L. This is possible Because already the licensed dendritic cells are activated. These licensed dendritic cells move on to activate further the T cytotoxic cells. So once the licensed dendritic cells move on to activate the native T cytotoxic cells, they do not need these CD40-40L mediated ligation.